Hello everyone, and welcome to FocalPlane Features. I'm Esperanza, the community manager of FocalPlane, the microscopy community site hosted by Journal of Cell Science. If you haven't heard about us, we created FocalPlane for you to connect with like-minded people and also find resources and information relating to microscopy. In our site, you can find, for example, news, interviews, posts, discussing tools or protocols, job listings, and also a calendar of events. Our community is free to access, and you just need to create your account in order to start posting your own contributions. So please check our website and then join our community. And today's webinar focuses on correlative light electron microscopy, and we are delighted to have three excellent speakers that will talk about the research of CLEM. So we'll have time for some questions after each of the talks. So please write your questions at any time in the Q&A box. And then remember that after the talks, we'll be also hosting a Meet the Speaker session where you'll have the chance to meet with the speakers in a more informal setting on remote. And now our first speaker is Yannick Schwab, team leader and head of electron microscopy core facility at the EMBL. He's one of the pioneers of correlative light electron microscopy, and his team is focused on developing new volume CLEM tools combining different imaging modalities. And today he'll talk about volume CLEM. So Yannick, the stage is yours. Let me stop sharing, and you can probably share your screen now.
All right. Thanks a lot, Esperanza. Thank you for the invitation. It's really a pleasure to share what we are doing in the lab, and it's even a greater pleasure to share the stage with Ori and Alexandra, who are very nice colleagues, and together we will try to cover the spectrum of scales in the correlative multimodal imaging field. So I'm pretty looking forward to their talks as well. I've changed a bit the title, but it's the same idea. I would like to introduce you to something that is not completely new. Maybe the name is a bit new. It's called CLEM and CLEM for correlative light and X-ray and electron microscopy, or just correlative X-ray and electron microscopy. And in the lab, we are trying to use this combination of modalities in order to really find the needle in the haystacks. And we are playing with different types of model organisms, multicellular model organisms. It's very often a challenge to find back the target, the bigger they are and the more complex they are. And we want to image these specimens with volume EM techniques, or some of the volume EM techniques, such as serial block phase, FIBSEM, or array tomography. I don't have time to go into many details here, but to illustrate the challenges here, I'm just showing you a data set by the FMI, which is a humongous data set. This is 7 weeks of acquisition. The amount of data that is here is absolutely impressive. You can spend of analyzing to so on and so forth. And of course, if you are interested in just cells, that would not be a proper strategy. A full organism of that, you would not have the throughput that's going to be large enough in order to the number of observations and so on. So for volume EM techniques, if you are not interested to image the full organism, it might be good to design strategies in order to target specific corners to then do the high resolution imaging. And this is what we are trying to develop in my team, and to play with different modalities in order to do that. The motivations are, as I just said, to target subvolume of interest, to optimize the acquisition time, and this as a consequence will lead to an increased experimental throughput, which then opens to the multiplication of observation and then hopefully lead to quantitation. What I'm going to show you today is what we are doing in FIBSEM, and the strategy is always the same regardless of the modalities that we are going to use. The idea is to build a 3D map of the specimen and then to define coordinates of the target. And by defining a set of moves or by trimming the specimens, we will drive the acquisition precisely to the position where the object is located. So I will show you some illustrations of the three modalities that we've been playing with. So light, to map the specimen, but also a combination of light and X-ray. And then I will finish by like CLEM per se, so you're just using X-rays to guide the target. But and the example I'm showing you is coming from a very nice paper that has been published recently by the group of Detlef Arendt, and the work is by Jake, who you see here. So Detlef's group is interested in the evolution of the nervous system, and one of the models that are looking at is the sponge, which might be surprising at a first glance to study the nervous system because sponge don't think that much. But indeed, Jake figured out by looking at transcriptomic data and other modalities that these sponges, they have a few cells that are scattered across the tissue that do have some neuronal characteristics. And by just looking at confocal microscopy, you can find back those putative neurons or neuroid cells hidden in coinocyte chambers. So these are the chambers that are making the filtration unit in the sponge. The problem is that not all coinocyte chambers do possess neuroid cells, and therefore you need some targeting, because in EM, if you want to image them, you cannot afford to go randomly. You would spend a lot of time to find back a cell of interest. So here we, in the lab, we developed a rather common strategy, which is to image in confocal the specimen, and then to prepare it for electron microscopy. And by just using the knowledge of the 3D position of the target within the block, and by overlaying the images, we try to trim the specimen in order to expose the region of interest to FIPSM imaging. And when I say we, of course, you can imagine that it's not myself. And here I would not acknowledge enough Nicole and Julia, who did the hard work for navigating into this specimen. So they did the trimming on this block because at this stage we didn't preserve the fluorescence in the resin block. It was completely opaque. Precise trimming, they managed to position a field of view in the FIPSM to image the full coinocyte chamber. So for one, it was interesting to have a look at the full coinocyte chamber to understand the cell's organization within the coinocyte chamber. But also, this targeting was not precise enough to decide to focus only on one cell type. So it was important to take quite a large field of view in order to compensate for potential mistargeting. But this was nice enough to recapitulate the full 3D organization of the coinocyte chamber. And after segmentation from the Kreshe group, we managed to realize how those neuroid cells that you see inside the volume of the coinocyte chamber do interact with the coinocytes themselves. And this is quite fascinating. You can also spend many, many hours looking at that to see how the coinocytes can tingle the neuroids that are hanging in the middle. The second illustration model correlations that I would like to show you now is something that is quite old, actually. That's something that we started very early on when I started to work at EMBL, which was to use fluorescence and X-ray in combination to target rare events. And this is a work that was performed by Mattia, a former postdoc in the lab, in collaboration with the groups of Jackie Goetz in Strasbourg and Frank Winkler in Heidelberg. And here the challenge was to image in the mouse brain, so this is intravarietal imaging, to then be able to target back single cancer cells that were trapped or hidden into the cortex. And the multimodal correlation that we designed for that had two advantages. So one was to find back a target, but the other one was also to be able to switch from live cell imaging to EM, and then compensate for the potential sample preparation artifacts and tissue deformations that are known to happen in these processes. And the strategy here is to image in the living mouse, then prep the samples for EM, there's deformation. But these samples, the resin-embedded tissue, we can then image with X-ray to find back some fiducials and then target the cell of interest here. The illustrations on how this works. So you have here multiphoton imaging in the living mouse, where we can find the target cell, a bit in the middle of the field here. And at the same time, we image also the vasculature, which is making a very nice 3D coordinate system. Prep for EM, you would recognize that we can image inside the tissue. We see also the capillaries here, which helps to reconstruct in 3D. I will play that here. The coordinate system, and a simple one can overlay the fluorescence onto the X-ray, which means that we position we place the position of the target cell within the topology of the resonant metal block. And with this knowledge, then a trained electron microscopist can precisely trim the specimen, as you see here in this drawing, to expose the cell of interest to FIB-SEM imaging. So here I don't go into the details of the FIB-SEM imaging, but just that important to position the field of view exactly where you need, because the field of view has to be right in order to go fast in the acquisition and to not be too overwhelmed with data. So here you see that Nicole and Mattia managed to find back this exact same cancer cell that has been imaged live into a living mouse. And then I will finish the presentation by showing you another application of correlations, but this time only with X-ray. And for that, I would like to introduce you very quickly to the group of Thomas Schneider, who is working in Hamburg with the synchrotron. And what they have developed there is fast X-ray imaging, which for us was very, very attractive. So, I mean, as you've seen, with X-ray, you can go into a rather thick specimen and you can image what is inside. But interestingly, in Thomas' group, the robotized limb that can handle the transport of the sample from the see here in this curve here to and they can go very fast also in the image acquisition, spending less than 5 minutes per sample, which opens to a very, very large throughput. So we were very excited by this ability to screen many specimens, but also the X-ray was much more powerful to image the details of our specimen inside. So you see here, different stages. We have another marine. The probably barely see on your side, but very important. We manage also to image spongilla and so on. We do have cellular resolution while keeping the context of the resin block, which is very important for the targeting. So this high throughput imaging methodologies has many attractive sides. One of them is to be able to screen for a large number of specimens, which is already very good for doing some phenotypic analysis. You can identify some cripples or some affected specimens with given conditions. But more importantly, for this correlation things that we are interested in today, is that you have the ability to screen for the most interesting sample, the individual that you want to image further downstream with much lower throughput. So one is high resolution X-ray imaging, and Alexandra is going to talk about that later on in half an hour. But for us, it was very interesting to build maps, 3D maps, to navigate into those samples in 3D. And you will understand why it was powerful for us. Remember the sponge? This is we are back to this example now, where we had to overlay confocal images more or less roughly onto the image of the resonant metal block. We took similar specimens to the synchrotron. We imaged them in two ways. So the first one with an X-ray, with a lab source, lab X-ray source, that topology of the resin block and of the tissue within the resin block. But then the same specimen was sent to Hamburg and imaged at higher resolution, where you can actually visualize the choanocyte chambers, and if we zoom digitally here, spot single neuroid cells inside the choanocyte chamber. And a volume EM expert in the lab, Paolo Ronchi, combined these two modalities, X-ray in the lab and high-resolution X-ray from the synchrotron, to do a precise targeting for FIB-SEM imaging. And what you can see here is a registration of the high-res synchrotron X-ray in green into the low-res lab source X-ray in gray. And that was very good now to precisely position the FIB-SEM sample prep, if you will, to image of interest. So this done in the choanocyte chamber. And that has the advantage to have a much smaller field of view which helps to increase the resolution. And in this time, we were able to dig into the characteristics of those cells to check if they indeed had some neuronal characteristics. So with this, I hope I managed to convince you that 3D mapping is very important when we want to do volume EM acquisitions in precise positions in rather complex specimens. And this is a way to quickly go to your target, which means that you can increase the experimental throughput and also optimize the data size. This kind of targeting is very efficient when you play with different imaging modalities, and therefore you can adapt to your assay, to your questions. And also to go faster, they include like a precision trimming step. So I don't have time to talk about that now, but in the lab now we are working exactly on this to see how we can improve the precision trimming step in order to go even faster in our targeting. So that's leading me to the outlook, that we want to be as automated as possible to go fast. And of course, the next challenge that we are starting to work on as well is to deal with the images and to extract relevant information from them. And this will, of course, come with image analysis. And with this, I'm done with my talk. And of course, I would like to acknowledge the many people I'm working with, and also and the collaborators, the project of whom I've shown some examples today. And thanks a lot for your attention, and happy to take your questions. And I should be sharing, I guess.
Thank you, Yannick. Let's see if some questions come in. Yeah, we have the first question by, I'm not going to pronounce properly the name, I'm sorry about that, but Brecher says, wonderful talk, how easy or hard is handling X-ray data sets and including it into CLEM approaches?
So from the lab sources, it's pretty easy, at least in our hands. But the tomograms that are coming from the synchrotron are a bit more complicated. And here we fully rely on the hands of experts. I guess Alexandra is going to talk about that because it's quite a challenge to reconstruct the tilt series at multiple distances and so on. These are very, very large data sets. And indeed, it is a challenge right now. So we have not reached the level of services that we want to reach in the next few years, together with Thomas Schneider and Liz Duke, actually, in Hamburg. So for now, we are still dealing with one that, if we want to expand that to like a high throughput imaging and cope with the amount of data, yes, indeed, this is something we have to tackle. Right now, it's done by hand and requires specialized scripts to reconstruct and so on. And then the data size can also be overwhelming. I hope it answers the question.
Okay, let's see if more questions come in. Waiting. We have another question from Leandro Lengruber. Both hydrophilic and hydrophobic resins work well with X-ray?
Okay, so I think you refer to epoxy resins and methacrylate resins like low-acryl. So we've tried them both in the synchrotron as well, and it's true that the low-acryl, they change color when you expose them to the X-ray. So I mean we've not tried to blow it, but I assume that yes, I mean if you expose them too long, you are going to burn your specimen. Even epoxy resin blocks, if you expose them too much, they turn black after X-ray imaging. So here again, I mean, I look at Alexandra. I think she can also answer this question because she has much more experience than me with X-ray imaging.
Yeah, we're also still exploring different types of resins. It's also, in our cases, we try to go nanoscale. We typically apply even higher doses. So it's, and also since you look at nanoscale, you also see, if anything, even very subtle changes. So you need something super stable. But I think it's a lot of matter of absorption, actually, because if we notice that if you use unstained tissue, for example, even if it's in paraffin wax, which you think, okay, it's smooshy, then it's much more stable. So it's also a matter of if you put heavy metals in to stain your tissue.
An interesting observation from what Paolo did in the lab, but this time with lab sources, so not a synchrotron X-ray, is that when you have embedded fluorescence in low acryl, it survives the exposure to the lab sources, which are much less bright than what they have in the synchrotron. So which means that if you do like a normal imaging, just to have the topology, not too high resolution and so on, it is still pretty okay, pretty safe to work even with low acryl.
Leandro also asked, let me see, because it's moving back.
Does en bloc staining interfere with the imaging? Yeah.
En bloc staining interferes with the imaging, yeah.
I think it helps with the X-ray imaging for sure. You have more contrast. But here again, I would just refer to Alexandra's talk. I think she's going to mention this. But yes, indeed, we observed that stained material gives a very nice contrast in the X-ray modality that we used. We tried also unstained specimens. You can see something. Actually, there are interesting behaviors. Some cells appear black, and I don't know where the contrast is from. But you still see the topology of the tissue. Yeah.
So let me just, there's one more question and then we'll just move to the next talk. So Sami asks, when moving from X-ray or IF to 3D EM, what tools do you use to reveal the ROI for FIB-SEM? Only ultramicrotome and diamond knives and coordinate maps?
Yes, absolutely. So we try to, yeah, absolutely. We try to target with a ultramicrotome, and for now, what we are using is the surface of the block as a reference to dig in. I've not talked about that today, but Paolo has published a paper last year where he uses, or he preserves the fluorescence inside the resin block, and then put the block in the confocal. And this is also a smart way to avoid the registration step that we need when we do a sequential imaging. And then he's adding near-infrared branding to put some landmarks on the resin block, which helps for the targeting in the microtome. But indeed, it's very efficient to use the microtome because it's rather fast to progress like through tens of microns inside the block and then to expose the region of interest. And it's precise. Ultramicrotomes are nanometric precise, so which is very good.
Perfect. Thanks, Yannick. Let's move to the next speaker, and then maybe we can have again open questions for everybody. So our next speaker is Ori Avinoam. He is a group leader at the Weizmann Institute of Science, where he studies the molecular mechanisms and physiological functions of membrane remodeling using advanced imaging techniques and multimodal imaging. And today he'll talk about room temperature versus cryo-CLEM.
Okay, am I audible and visible?
Yeah, all good.
I can only see my presentation now.
You're good to go.
So good afternoon, everyone. Thank you so much for joining. I have to start with a confession and a bit to lower the expectation. When Yannick invited me to this forum, I thought, oh, that would be an excellent opportunity to think about compromises between room temperature and cryo-CLEM, because I think that is of very high interest. However, I'm afraid I did not anticipate that I would have to do it in 15 minutes. So I had to keep it quite conceptual, and I'm hoping that we can have a discussion afterwards or after all the talks. So I'd like to start with the facts, or of the obvious sort of definition of how we often view CLEM. And that is that on the one hand, we have fluorescence microscopy that benefits very much from molecular specificity, meaning we can label molecules brightly over a background. and they benefit from the ability to collect dynamic information. However, they suffer a lot in their ability to produce a comprehensive view of the specimen, meaning what we label is what we see, and we are limited in the number of fluorophores even. And also it is limited in resolution, even when we think about super resolution techniques now spanning into the tens of nanometers, it doesn't reach the high resolution of the electron microscopy. And you can immediately appreciate that electron microscopy suffers exactly where fluorescent microscopy suffers and benefits here. So almost naively, we would imagine that connecting these two modalities would lead to this super method. However, as in everything in life, everything comes with a trade-off. And I'd like to sort of discuss some of these trade-offs with the following example. So if we were to study the organisms, for example, that Yannick were referring to using live imaging, we would normally turn to confocal microscopy, right? Because it enjoys very good relatively very good temporal resolution. It can image relatively large specimens. However, we pay for acquiring 3D data sets by using the temporal dimension to acquire many images over time. And also we need time to acquire the fluorescence. We need time to register the signal over the noise. Therefore, confocal microscopy is always going to be limited in both spatial and temporal resolution. If we want to compensate for that, one great way to do that across modalities is to reduce the dimensionality, going from a 3D to a semi-2D imaging modality like total internal reflection fluorescence microscopy that only images about 100 or 200 nanometer close to the base of the cell. So, for example, an adherent cell on the cover slip. And while this offers now single molecule fluorescence in terms of the ability to resolve and also very high dynamic information, there is zero spatial information. I mean, we are completely diffraction limited. If one wanted to advance, you could combine, for example, turf with a structural illumination. And I don't know if you can see that, but now the circles or the blobs turn into a type of circles. So you can see that there is some sort of spatial information, but maybe not enough to compensate, to even increase the spatial resolution. Now we have to completely remove the temporal dimension. We have to look at fixed images. And now we have enough time to acquire lots and lots of data, for example, using turf and storm to get an additional information in the third dimension. Again, approximately two-dimensional, but still recovering some three-dimensional information. Now, if we wanted to go beyond, so, you know, we had something, then we had a blob, then we had a circle, now we have a semicircle. So actually the circles we were looking at before is the aperture of this open structure. But in order to really interpret what we see, we need a high resolution of electron microscopy that can reveal that this beautiful fluorescence actually comes from a proteinaceous coat that is assembled on a membrane in the process of invagination. And if we wanted to go even farther, now we can step into the realm of cryo-transmission electron microscopy, where again, in this particular example, I'm showing you a thin specimen, an isolated vesicle in solution in a thin film of water, and you can appreciate that now contrast looks completely different, while here we have mechanisms or dyes to enhance the contrast, such as heavy metals. In cryo, we typically look at the native contrast that simply comes from the molecules themselves. And by adding along, by the way, all of these approaches, a type of computational averaging approach specifically for cryo, one can obtain also structural information as to the organization, for example, of this protein. And I would say that you could also resolve the ultrastructural organization of the protein, either, again, by an averaging approach or by simple segmentation. So what becomes immediately apparent when we look at multimodal imaging is that they are full of compromises. The most trivial one is between the temporal, high temporal resolution of fluorescence microscopy to the high spatial resolution of electron microscopy, but also there is the tendency to go for more in vivo, more native samples to a more in vitro, the more resolution we need. And also really, as Yannick touched beautifully on, the matter of throughput. While fluorescence microscopy is still relatively a high throughput method, towards electron microscopy, and the more complex combination of modalities we want, the lower the throughput. And that can, and so in correlative microscopy, what we try to do is actually get a much better compromise between all of these parameters that are inherent trade-offs of combination. And what I will try to do next is somehow touch upon what would be the typical compromises between cryo and room temperature CLEM. But just to summarize, so we have this food on the way, and I think all of these things were on Yannick's last slide, right? Anything that we want, for example, if we want the highest temporal resolution, we will be moving farther away on all of these axes from these other 5 elements. So for example, the dimensionality, the volume, the more volume we need, the more we have to compromise, the more throughput, and the closest to the native state. So that would be a neuron firing in my brain, or attempting to do that, or a neuron firing on the cell culture dish, or a vesicle from that neuron, or a protein involved in the process of neurotransmission. Now, when it comes to correlative microscopy, when one engages in such an endeavor, the first question that comes to mind is, what do you need to see? And the reason is that really helps set us on the right course for the experiment. It sets the essentials. So for example, it differs a lot if we want to see a cell, or a cell in an organism, like we just saw, or if we want to image organelles, or something on the order of 100 nanometer, like a vesicle, or a virus that might be the reason why we are all meeting over Zoom, or even a protein. Because as soon as we define what we need to see, it defines the endpoint modality, whether we could use a scanning electron microscopy approach, a transmission electron microscopy approach, and very often it will tell us whether already we need to be operating either at room temperature or under cryo temperature. And once we know the modality, we actually also know the way, or the many possible ways to reach those modalities. And I think the textbooks are now full of both cartoon and also detailed protocols how one might proceed to any of these modalities. So by just defining what it is we need to see, we already sort of know where we land at the end. And the other reason it's important to define what one wants to see is that very often defines where the fluorescence microscopy information, or any other dimensional X-ray, whatever else needs to be combined on the way, needs to be involved. It needs to be incorporated. For example, we can image the samples still living, still alive, but then we recover actually full dynamic information. But while we proceed down this track, things may still move. So we suffer both in our ability to correlate, in our ability to find back the object, and sometimes in the interpretation. But fluorescence microscopy can also be incorporated post-fixation. So now we, again, as in the examples, we give up on the movement in the sample in order to recover both on the spatial resolution and in the precision at which we can correlate. And I will give you an example in the next slide. And further, we can also incorporate the fluorescence microscopy even closer to when we image, perhaps after thinning down cells into lamellae, or sectioning them either in cryo or room temp, et cetera, et cetera. So what I'd like to offer is that the most important question to start with is, what do you need to see, even before you consider what are the compromises that you will make, because the compromises are very often inherent. So let's look at one example. This is a workflow that goes from cryofixation, but then into resin embedding, and the imaging will be done post-processing in order to visualize something on the order of 100 nanometer and with very high correlation precision. So we start with the living cell in the dish, we cryo-immobilize it by high pressure freezing, and then it goes through resin embedding, trimming and sectioning, and the imaging is done on the resin sections. So you can see that both red and green fluorescence is nicely preserved, and these multispectral beads in the sample are actually the multifluorescent spots, I should say, belong to beads that we can add exogenously on top of these sections in order to create a registration system between the EM and the fluorescence modality, and I'll demonstrate it in a second. Let's just zoom in on one area so you can appreciate that even though fluorescence microscopy offers these very huge fields of view, still for the EM, we only actually hone in on very small fields of view, as Yannick actually alluded to. But what we can do here is, because we have this very high precision, I mean, the fluorescence microscopy is done on the sample after it has undergone all these preparatory steps, we can get very high correlation to nail down these pits. And then we can, sorry, this is my explanation for the beads. So if we have the beads some fluorescent beads that are also electron dense, and we have some other organelle or feature that we want to localize, by registering those, we can align the two the beads and get an alignment of the object of interest. So once we target, we use the fluorescence microscopy for targeting, which increases the throughput by quite a bit. We can combine this with a 3D technique like electron tomography, for example. And what this kind of information gives us is quite high contrast. And what I hope you can appreciate is that with this level of information, you can easily segment, for example, the membrane. So if you're not used to seeing these structures, this is a membrane with its bilayer, and this is a protein coat on top of that membrane. And where the precision comes into play, why it is important to acquire the fluorescence information after all these preparatory steps and movement is because in this particular case, we wanted to ask whether the red labeled protein and the green labeled protein are localizing differently along this pit. And by acquiring a lot of data, so we have sufficient throughput, we can calculate the probability of where these fluorescent signals localize with a precision that is better even than 100 nanometers. And then you can see quite clearly that the red protein, which was actually a protein component of this coat, centers more or less exactly where the coat appears in the EM, but the other molecule localizes more towards the neck of this membrane invagination. And so this is one example where correlation precision is extremely critical and would only be attainable in a regime that combines the fluorescence microscopy after sample prep. How does this relate to cryo? Well, cryo appears to be a wonderful modality because it eliminates all of these extra sample preparation techniques, and the imaging can be now beautifully done again right before the sample enters into the electron microscope. However, now we are limited both on the fluorescence microscopy side to microscopes that are specialized, that have a cryo stage and are able to navigate. And in the absence, the current absence of any kind of immersion media, actually we suffer also on the level of resolution. So what you see in here in the fluorescence microscopy, again, if you're not used to it, this is an EM grid with some cells that have green fluorescence. And once we image this basically entire grid, we can do the same with the electron microscope. Also, of course, a typical compromise for a cryo workflow is that it requires cryo electron microscopes. And then we can target specific cells. We use the same concept of beads. In this case, they are adsorbed to the surface. And then we can target with high precision events that happen within these cells. So in this particular example, the cells are intact. We are imaging them using transmission electron microscopy at the very thin lamellar regions of the cell. But this could easily be, or I don't know if easily, but could now very well be combined also with a thinning step before the imaging and after the imaging. And just to show you, then you can combine this the same with tomography, and the typical compromise there is on the contrast itself. Okay, I don't know how I'm doing for time, so I'll go over the next part extremely quickly and then we can discuss it. So just to point out that the same principles now apply to SEM. But SEM adds a complexity, as we just saw, because it's intended for high fields of view on the expense of spatial resolution. But now we gain the third dimension, and the third dimension complicates things both for the correlation because we need to correlate in 3D, and also for where to acquire the fluorescence microscopy. So I think Yannick gave a better example than I'm going to show, but just to show you that things more or less work the same. We can, the cells or any kind of tissue can be harvested live, high pressure frozen, embedded, and then after polishing or trimming the blocks, we can acquire fluorescence microscopy maps, target specific regions of interest. However, because here it's complicated to introduce any kind of fiducial system because this tissue is still embedded within the resin, the correlation precision that we could achieve is only sufficient to target relatively large object like this vesicle that is co-labeled here with red and green. But still, this is very good if one needs to acquire large objects with the entire 3D information intact. Another clear advantage of this technique is that resolution, you can acquire these volumes with isotropic resolution, what you cannot do in 3D transmission electron microscopy, unless you do serial sections, but that has other limitations. Lastly, I just want to point out that this is also possible to do in cryo, albeit less frequently done. So here I go back to the example of cells grown on grids. Well, I've already shown you fluorescence. This is how the same would look in a cryo SEM. In this case, correlation can be done in 3D because we can label something in the cell, like the lipid droplets shown here in green. So they provide us a type of 3D coordinate system to target. And surprisingly, contrast in cryo-FibSem is not that bad, especially of membranes. So it's definitely a modality to consider, especially if your object of interest requires observation closer to the native state, where you cannot embed it even in resins. Okay, so with that, I hope I am on time and I was somewhat coherent. And I'd just like to thank my lab and collaborators, especially Nadav Sher, a PhD student in the lab that helps me a lot in thinking about these things and with illustrations and experiments, and he's excellent. And with that, I'm happy to take any questions.
Thank you, Ori. That was a great overview of all the techniques. We have one question already from Sami. What microscope was used to collect the cryo-FibSem data?
That was the Zeiss crossbeam. I'm just going to stop the share or I don't see. So we use the Zeiss crossbeam 550 to acquire that.
Let me see if there are any other questions. While we're waiting, I was wondering, like, in terms of like if you're doing immunolabeling, would that be different? Like, can you use, let's say, the same antibody would work in one technique versus like just TEM versus cryo-EM, or
so the
epitope masking and things like that?
The challenge always with introducing antibodies is the same as introducing, for example, fiducial markers. You need to permeabilize the cells, and there you have to balance between damaging or changing or intervening with the ultrastructure and the outcome. Now, there are protocols that use gentle detergent to introduce these antibodies, and to a certain resolution, they work great. But the more resolution you acquire, the more you see the effects that you created. So it's a bit like what Alexandra said before, that you know, when you go high resolution, then you have to keep your samples closer to their native state.
Let's see if there are any other questions. Nothing comes in. So I'm thinking in terms of timing, maybe we can go with Alexandra, and then we can continue with more questions afterwards for all the speakers. So now is our last speaker today is Alexandra Pacureanu. She is a scientist at the European Synchrotron, where she leads the nanoscale X-ray neuroimaging unit. Her team works on developing cryogenic X-ray phase contrast nanotomography for 3D imaging of thick biological tissues. And today she'll talk about multimodal volume microscopy to explore the structure, function, and elemental composition of tissues and cells. So Alexandra, stage is yours.
Thanks so much for the introduction. I seem to have some issues with sharing my screen. I'll try to share just the window. Let's see if it can work. Can you see it full screen?
No, not
because
now.
also yesterday was not
Right.
okay.
Maybe if you try to first do the full screen and then share that full screen, maybe that might work.
Yeah, if I do full screen, I can't access the. I'll try once more to see. I'm not sure why. The share button is not active when I try to share entire screen for some reason. Oh well. Worst case, we try. I share just because otherwise you see my slides but not in full screen, right?
Yeah.
It's not so nice, but at least we. Now I can't see the window even. Okay. Okay, let's try like this. So I try to zoom in as much as possible. Do you see something fairly reasonable or not? Really?
Yeah. That should work.
Perfect. Okay, sorry about that.
Mm-hmm.
So yeah, so I thought I was going to be a bit the black sheep here in the CLEM community, since my main interest is more X-ray microscopy. But Yannick already helped me to feel welcome. So I put a bit in the middle here of the techniques, X-ray holography in 3D and 2D, which is my main interest. But we've been exploring to combine it with all kinds of modality, from cryolight microscopy to super resolution microscopy to EM and so on. And why we do this? Because of course we want to access different types of information, from structure at multiple scales to chemical composition and molecular information, in order to be happy and obtain the knowledge we are looking for. So just to give you a brief introduction of the core techniques that I'm working with. So on one hand, it's X-ray phase contrast imaging, in particular holography. So this we combine with tomography, so it's a 3D technique. And what we obtain is the distribution of the electron density in the sample, so the structure basically in 3D. And this can be combined quite easily in the instrument that we've been developing with X-ray fluorescence. So this is different from visible light fluorescence. In this case, we don't need to add any label. Typically, we work with vitrified samples which haven't been treated in any way. And we raster scan the sample with the X-ray beam, and then we record spectra for each point, which we can fit, and then you can tell apart maps of distribution of different native elements. In our particular cases, with atomic numbers higher than 13, due to the X-ray energy that we use for excitation, but with very high sensitivity, so sub-ppm level sensitivity. And this can be also done in 3D, but it takes quite a lot of time, so we do it less often. So in particular, the holography technique, is quite versatile to be combined with all types of other microscopy modalities. And it has a few advantages in the sense that it's quite easy to acquire multi-scale data by simply moving your sample between the X-ray focus and the detector into a divergent beam. And it's a full field and fairly rapid imaging technique, and radiation dose efficient compared to other technologies with X-ray imaging. And it offers exceptional contrast in also in unstained cells and tissues. This is just a picture of the instrument. So inside here, we have combined the X-ray holography, the X-ray fluorescence, and there's also visible light inline microscope. So basically the sample can be either in room temperature or vitrified, and is placed in this chamber on a rotational sample stage, and then it can be imaged with both the technologies that I mentioned. So just to give you an example, so what kind of measurements one can obtain. If we take a single cell level, on one hand, we can obtain maps of different elements. So unfortunately, my movies are not going to work. So here for phosphorus and zinc, there are some movies, but I'm afraid I can't make them more because they're GIFs actually. But yeah, I can show you later the movies if you want to see. But just to give you the overall idea, so one can use phase contrast to segment at least some of the organelles, not all of them, and then correlate the X-ray fluorescence map. So each map of chemical elements, and then extract, for example, molar concentrations in different organelles, and then what kind of questions one can address with this. For example, the toxicity of nanowires. So in this case, people want to see how skin cells react to silver nanowires, which are as thin as 33 nanometers in diameter. So they've been sprinkled on top of cells in a living culture, and then left for a little while, and then the cells have been vitrified, and then we looked in 3D with phase contrast to see whether the wires actually go inside the cell, and also to see how the cell reacts. And we could see that there's both a mechanical reaction in the sense that the cell manages to kind of fold and crumple the cells and encompass them in endosomes, and on the other hand is trying to attack the invaders with sulfur and copper. So we saw deposition of sulfur and copper on the nanowires, which we could nicely see in the silver channel. Right, but there's also a little bit of light. So this is something that's still a bit of work in progress, but is now advancing quite well. So I should maybe mention that this is an instrument at a beamline, at a synchrotron. So it's possible to, if you have an idea and you think you need all these techniques, it's possible to submit proposal or to contact people to work on a collaborative basis. So in this case, the idea is to combine cryo light microscopy with X-ray fluorescence and maybe X-ray phase contrast. So one has to go from either plunge freezing or high pressure freezing plus cryo ultramicrotomy to a system that we have now also in a lab close to the end station, where we can we have the X-ray microscope, and there's a tool in place to help for guiding the imaging, so one can find the targeted cells. These are the very first images acquired. So it's these are the fluorescence, visible light fluorescence images, phase, 2D phase, and X-ray fluorescence of zinc, potassium, and selenium. There are some selenium nanoparticles. In the meantime, there's been progress, so there's nicer data, and my colleagues are working on preparing a publication and also making things a bit easier to use on a regular level. And now just showing like a few more on the tissue level, like what can it be used for, what kind of questions can be addressed by combining these two X-ray techniques already. So in the level of brain tissue, for example, one can try to look at structure. So with in this case, it's unstained tissue, and with the phase contrast, we can see, for example, in a model of Alzheimer's, so it's a mouse Alzheimer model, we can see some of the neurons which are dead or dying simply because they become denser. So they have a different contrast and a different shape. And then we can take out a slice from this sample after imaging, and we look with X-ray fluorescence, and then we can see the distribution of different elements which can be relevant for Alzheimer's, in particular iron. So you see on the top row is neurodegenerated cells and the bottom healthy neurons. And we can see in particular the iron that seems to concentrate, but in the cell, which of course it's becoming smaller after it died, but also it seems to be a bit in a higher quantity. And similarly, this can be done in different other types of tissues. So this is lung and heart tissue, which is also unstained, and one can combine the structural information with the chemical composition information. And because it's X-rays and they can penetrate through heart tissue, one can also investigate mineralized tissues. So in this case, we took a sample of bone, and we could look at quite high resolution. So it's a voxel size of 25 nanometers with the phase contrast. And then we did also 3D fluorescence imaging, and we could see, for example, if we look at the 3D rendering of reconstructed zinc and calcium, we can see here a structure which looks like a part of a cell and dendrites. So this is actually the neighboring wall of an osteocytes, which is a cell in the bone matrix, which was not actually visible with in the phase contrast, because you can't see the cell. It's actually outside the sample here, but just because of this distribution of zinc around the cells, we can see this. And so this can be, of course, applied to any kind of samples from our ecosystem. So recently we looked also into pollen grain, and by combining in cryo conditions this time the phase contrast and the X-ray fluorescence, we could see that the exine, so the outer wall of the grain, it's actually a nanofoam, which is like so far there are not any known bio nanofoam at this kind of scale. Right. So coming back to the brain, since it's one of my main interests. So we also combine X-ray tomography with 3D electron microscopy, and this can be interested in the sense that with X-rays one can cover larger volumes and reconstruct, for example, neuromorphologies, dendrites. But then in order to look at synaptic connections, one may need to use higher resolution. So in this case, volume TEM. So one can target a smaller region and then combine the information of long-range dendrites and local synaptic connection and quantify this. And then even better, so this is work spearheaded by Carles Bosch at the Creek. We would like to go from in vivo neurophysiology through all the way through synaptic level comprehensive circuit reconstruction. So in this case, starting with 2-photon microscopy in vivo, then also ex vivo, and then going to synchrotron micro-CT, where a quite large sample, so here an olfactory bulb can be imaged, and one can trace a specific structure which has been labeled with a genetic label, and then try to register this all the way to 3D EM data. And within this process, only for the X-ray microscopy part, we can do it at multiple scales. So starting from around 1 micron resolution, where already the cell bodies and some dendrites can be traced, to zooming in to the 100 nanometer level and tens of nanometer level, where one can start to detect the first synapses, and we keep pushing the resolution for X-ray microscopy to try to make it possible to investigate samples simultaneously at very high resolution and at quite large scale, so without cutting them very small, or with a throughput that allows you to cover very large volumes. So I'll stop here with some pictures from the snow. So what we have around here in Grenoble at the moment. Sorry about the bug with sharing my screen. It was a little bit less smooth than I was hoping, but I hope it was still useful. So I'll stop sharing. And
Okay. It was clear, and we could see definitely the images. So
Thanks.
that was good. Let's see if some questions come in. In the meantime, there's one here. So Sami asked, how were patient samples collected and preserved? For example, the Alzheimer's brain samples for the X-ray nano-CT fluorescence. And do you use the same instrument for both imaging modalities?
Yeah, so for the Alzheimer brain, that was a mouse brain actually, but it's most of the times the samples are dehydrated and embedded in paraffin. So it's a typical preparation that people do for histology or at the hospital. So it's unstained. And then, yes, so the X-ray nano-CT and the X-ray fluorescence are done in the same instrument, this kind of submarine. So the sample is placed on the same sample stage, and then we can switch between the two quite easily, the two techniques.
Let's see. In the meantime, Ori, we have two new questions for you that came after we started with Alexandra's. So it's actually a second question from Sami that asked before. So what detectors were used for cryo-FIB-SEM imaging? Very nice contrast. What is the voxel resolution and limitations to cryo-FIB-SEM imaging on the Crossbeam 550?
So, okay, these are a lot of questions. I should point out that we recently published this as a manuscript in iScience. You can look, Nadav that I mentioned is the first author, so all the details, and there will also be a protocol published soon with all of these details, so you don't have to remember. Starting from the end, of course, stability is always more challenging under cryo conditions, keeping the milling and the imaging consistent. Voxel resolution is tunable on a FIB-SEM for these because we wanted relatively, again, large fields of view. If I'm not mistaken, we imaged at 30 nanometer cubed, but I might be specifically wrong here. And the contrast mechanism is not really elucidated, I have to say. Obviously, if there is no topography, it's a bit of a and there is no stain, it's a bit of a mystery. The current theory is that it comes from surface potential that builds and membranes generate apparently quite a lot of that potential, or the interactions of the membranes and the electrons. And the last, what detectors? So you use a backscatter detector, but we use both detectors. One to see if there are any artifacts, because it shows the artifacts better, and then the other one is actually for the imaging. So we acquire both, and we either use one or combine them to one image. I hope that answers.
We have a second question for you, Ori, from Leandro. How much investment in time you would say to a person before going into cryo-CLEM? For example, a few months?
Again, the question is, it depends what you need to see. My typical philosophy is that people should be, you can, in 3 months should be completely independent. And I've never seen anybody struggle beyond that. I mean, normally people already get data way before, but to become fully independent in these experiments, probably 3 months is a sort of a rule of thumb. And of course, you can elaborate in the discussion exactly what that means.
Yeah, so I don't think there's any other questions right now. So I would say all these more like specific questions, definitely something that we can discuss now in the after session with the mid speaker at Remo. So before we move into that setting, I just let's wrap up here. So thanks again, all the speakers, for the really interesting talks. We're going to move now, as I said, to the mid speaker sessions, where you're going to have the chance to speak with them. So when we switch to the room, you're going to find yourself sitting at a table. So you just have to click on an empty chair, and then you can move between tables and join the other tables. So if you don't want to attend this session, just make sure that you close the browser window for leaving the event. Otherwise, you're going to be just appearing in there, but not really interacting with anybody. So thank you all for joining, and I hope to see you all in the after meeting. See you there.